Today I want to talk to you about some work we've been doing, and I want to take you back to the beginning about embryonic stem cells. And I want to give you a spoiler alert by telling you now that mouse embryonic stem cells and human embryonic stem cells are not the same. Okay? And this will become clearer as we work through the talk. I also want to finish by talking about some of the work we've been doing in the pig in this area. So mouse ES cells, we know they can differentiate to all the cell types found in the body, including the germ cells. In other words, they are pluripotent. And they were first isolated back in 1988, and this slide just is a cartoon which basically shows the procedures. So we're talking about isolating cells now from the epiblast, which forms following the differentiation of the inner cell mass into the epiblast and the hyperblast. The epiblast gives rise to the fetus and the extraembryonic membranes. So in the mouse, this is around day 4.5, just before implantation. Okay? So that's how the mouse ES cells were first isolated. Evans and co. were looking for the equivalent in the embryo of carcinoma cells, which they've been playing around with for a while. So how do we know that mouse embryonic stem cells are pluripotent? Well, if we take embryonic stem cells and we inject them back into the developing blastocyst, we can produce a chimera. In other words, a mouse that consists of the two cell types. The cells from the inner cell mass proper, or epiblast proper, and the injected cells. And this is a powerful technique which we've used to do gene knockouts to look at gene function over the last 20 years. And if we mate those animals on with a single mutation, we get animals with a double knockout, for example. And if we see some sort of phenotype, we start to understand what the gene does. So a very powerful technology that's been around for a long time. However, people have tried to isolate embryonic stem cells in other species, such as the pig, for example, and have invariably failed. So was there something special about mouse embryonic stem cells, in particular the 129 strain, or is it are these cells, can we isolate these cells from all species? So human embryonic stem cells were isolated in 1998 by Jamie Thompson's lab in Wisconsin. So there was a lot of interest in this because here for the first time was the possibility or potential for an effective treatment for a range of diseases and injuries which we can't cure. And most of you in the audience would know these examples. We typically talk about Parkinson's, type 1 diabetes, heart muscle, and also injuries to the spinal cord. And the company who produced this slide, called Geron, was the first company to go to have a clinical trial in the area of spinal injury. Now, I gave the spoiler alert that mouse embryonic stem cells and human embryonic stem cells are not the same. If we look at the mouse, then basically embryonic stem cells are isolated back here. Whoops. Prior to implantation. Post-implantation, we see a group of cells called epiblast stem cells. And a number of groups now have shown that human ES cells approximate to these cells in the mouse that are found in the developing embryo post-implantation. So typically we talk about mouse embryonic stem cells as being naive, and we talk about human ES cells being primed because they approximate to the mouse epiblast. So what's the difference? Well, I've told you before that mouse embryonic stem cells can make chimeras if we inject them back into the developing blastocyst. If we take mouse epiblast stem cells, which approximate to the human ES cell type, they don't make chimeras. So based on what I've told you, you can conclude that they're not as pluripotent. So what are the differences? Well, we've talked about naive cells being more pluripotent because in the mouse, the equivalent does not make chimeras. They seem to be more, naive embryonic stem cells seem to be more homogeneous. And this is a comparison now in humans, sorry, I should have mentioned before. More characteristically stable, more proliferative, better replicate embryo development. When we make teratomas, for example, with existing human ES cells, the primed human ES cells are out there, what we tend to see is an overrepresentation of the mesoderm germ line. But the caveat here is that we've only had, up until recently, one naive human ES cell to play with. And this was work done by Carol Ware back in 2014. In contrast, if you go on the NIH embryonic stem cell registry, you'll find over 300 primed human ES cell lines which we can play with. So in terms of their advantages or disadvantages, you'd have to say that the jury is still out in terms of naive versus primed human ES cells. Nevertheless, there are products out there, and the two that I'll mention today, Viaside, a company in the US, have a product in phase 1-2 trials for type 1 diabetes, and here they've taken ES cells, differentiated part way to insulin-producing cells or beta cells, and wrapped it up in this encapsulation device, and it's now in safety trials in the US. The other one you might have heard of is companies like ACT are looking at macular degeneration, and here they're differentiating human ES cells to retinal epithelial cells which sit at the back of the eye which process light information. And the advantage here is that the eye is an immune-privileged site, so when you start putting cells back into people, they don't tend not to be rejected if you target the eye. So I've talked now about naive human ES cells and primed human ES cells. Now I want to take you back further in development to a stage in embryo development shown here for the mouse, where the inner cell mass is yet to differentiate into the epiblast and hypoblast. So mouse ES cells were isolated from the epiblast. The human equivalent seems to sit further down here, post-implantation, and similar to mouse epiblast stem cells. Now, for various reasons, we looked at the pig. We went back and thought we'd like to try and isolate ES cells in pigs. Other people had tried, so we basically went back to the black drawing board and started again. And I had some very smart people in my lab, and they said, well, we know these cells are more pluripotent than these cells, so let's try and isolate ES cells from this stage in embryo development in the pig. That's basically what they did. To cut a long story short, we used in vitro produced embryos. We get the ovaries from the abattoir, mature the oocytes in vitro, fertilise them and grow them up to the blastocyst stage. So we take these whole embryos, we squash them down on the mouse feeder layers, basically because we don't want the inner cell mass to differentiate any further into the epiblast and hypoblast. And we use serum replacement instead of fetal calf because fetal calf serum tends to promote differentiation. That's not what we wanted. We all did this under low oxygen. And what we ended up with was a homogeneous outgrowth of pluripotent cells, shown here. So we didn't end up typically with a mixture of cells like you do in mice. We choose ES cells and then just passage them to clean them up to give you a homogeneous culture. Basically, the trophectoderm cells degenerated, leaving us with the inner cell mass cells, which became ES cells. If we wait until the inner cell mass differentiates, we end up with a mixture of cells just like you do in mice. So basically what we were doing there, we're isolating these cells at an earlier stage in development than had been used in the past in the mouse. So these cells behave like ES cells. They express OCT4 and NOG, and interestingly they express SSEA1, stage-specific embryo antigen 1, which is now seen as a marker for naive ES cells, as opposed to prime. They maintain their pluripotency. This immunohistochemistry was done at Passage 10. They maintain their karyotype. They can be vitrified repeatedly, and they really differentiate to cell types representative of all three germ layers. And we've also isolated this same cell type now in mice and cattle. And we've differentiated these cells, like I said, to other cell types. And this just shows the pig equivalent of retinal epithelial cells, which I talked to you about before, being used, currently trialled for macular degeneration in humans, which is that picture there. So, do these cells make chimeras? Because at the end of the day, this is the gold standard in terms of demonstrating pluripotency. Well, to cut a long story short, we produced chimeric pigs, and we were surprised to see this, but you can see some mottling along the back here because one of the boar that we actually used to fertilise the embryos we isolated our ES cells from was a Hampshire, which has a black coat colour. Now, white is dominant in pigs, so we didn't expect to see this mottling coming through because these cells were then injected to what essentially were white embryos. We haven't demonstrated germline transmission yet because that cell line was female, and female cell lines tend not to go germline, but I don't have time to talk to you about that. So what does all this mean? Well, we believe we've isolated a pluripotent cell type earlier in embryo development that's been compared with those used previously. We don't know if it's the same as a naive mouse ES cell. We're still yet to characterise these cells fully. Do they have added advantages for cell-based therapies? Well, I talked to you before about naive versus primed, and that naive are probably better when it comes to doing cell therapies, although the jury is still out on that one. It would be nice to isolate this cell type in humans, and we're looking to do that if we get some funding. But what about pig ES cells? Do they have a place in this story as well? Well, I think it's one thing we don't have at the moment is a large animal model for human ES cell studies. We have lots of mice ES cells from permissive and non-permissive strains, but we don't have a large animal model. So with the ability to make lots of embryos in the lab, we can look at basic questions about oxygen tension, for example. How easy is it to isolate ES cell lines under 5% versus 20% oxygen, for example. And we have this applied facility, if you like, where we can start to do the sorts of cell-based therapies in a large animal model for the first time. And there are plenty of pig models out there for things like blindness and vascular disease and so forth. So what I wanted to tell you today is basically that mouse ES cells are not the same as human ES cells, and that we believe that pluripotency is lost across development as the embryo develops. We believe the further back you go, the more pluripotent, perhaps even totipotent the ES cell. And I just leave you with a thought by Carol Ware, who isolated one of the first naive human ES cell lines now a couple of years ago. In a review she wrote that she believed that the full potential of ES cell-based therapies may only be realised if naive ES cells or perhaps something similar are used. Thank you very much.